In part 8 of our discussion of Chapter 4, we are going to pick up our conversation here talking about protein folding, but now we are going to look at enzymes that help to facilitate protein folding, chaperones. In previous lectures, I emphasized the order of events related to protein folding. So how do you go from a newly synthesized unstructured protein to a protein that is now structured? When we talk about that, we talk about secondary structure formation followed by tertiary structure formation. But what we don't really talk about there is the fact that for many larger proteins, this cannot happen unassisted. You need a little bit of help to make this possible. That's where chaperones come in. So that's going to be the focus of this recorded lecture, part 8. Now we're going to turn our attention to protein folding. As I go through this discussion, I'm first going to focus on the problems that can arise during protein folding, and then we're going to talk about the role that molecular chaperones play in solving that problem. First, let me just tell you how things would go normally. We've talked about this in previous lectures, but here I want to remind you of that. So imagine you have a protein here. This protein is newly synthesized, or for whatever reason, whether it be thermal stress, chemical stress, has become denatured. It is largely unstructured. Once that stress is removed, or for the case of the protein that is newly synthesized, it's going to start sampling different conformations. And this sampling process is going to lead down that protein folding funnel that we talked about last time. Remember, in the unstructured state, your energy, your free energy is quite high. And so you start sampling different conformations that incrementally lead down the folding funnel towards the lowest energy state, that is the state that corresponds to the native protein. So the goal is to take your unstructured protein, start sampling different conformations that lead to the formation of folding intermediates, and hopefully you find your way to that lowest energy state, the native protein. Okay, so now that's how things would normally go. But let's focus for a moment on what happens when things don't go that way. Well, a very big problem area here is centered on the folding intermediates. Before I tell you, let me remind you, the native protein, in most cases, at least the cases that we are going to focus on here, the native protein is going to be a globular shape. It's going to be roughly spherical. That sphere, that globular three-dimensional structure, it is going to have nonpolar materials on the protein interior, and it's going to have a hydrophilic exterior. So remember, amino acids like alanine, leucine, isoleucine, they have nonpolar functional groups. They are classified as being nonpolar molecules. So for that reason, they don't really want to interact with water. Remember, the suspension of a nonpolar molecule in water comes with an entropic cost. So in order to minimize that cost, globular proteins bury that nonpolar material away from water. We make the nonpolar interior of the protein structure largely solvent inaccessible, so you minimize that entropic cost. Now, that description is true for the native fully folded protein. That description is not always true for protein folding intermediates, such as the one shown here. So in those cases, you may have nonpolar functional groups that are still solvent accessible, but they won't be once those proteins find their way to the native conformation. However, during the intermediate's lifetime, while it is still in existence, while it's populated, those solvent-exposed nonpolar functional groups may create these nonpolar surfaces on folding intermediates. This is problematic because those nonpolar surfaces may bump up against other folding intermediates, and due to the hydrophobic effect, may start to promote the formation of aggregate structures. Last time, I talked about the protein folding funnel. And I told you that sometimes you will run into these local energy minima that allow for the stabilization of folding intermediates or misfolded proteins that have these exposed nonpolar surfaces. So in those energy minima, you can see very stable aggregate structures form, and these aggregates most often are very toxic to cells. Okay, so this is the problem. We want to address and prevent the formation of aggregate structures derived from these folding intermediates. So now we turn our attention to the role of molecular chaperones. What are they and what do they do? The molecular chaperone is simply an enzyme that is going to recognize folding intermediates, aggregates, or just misfolded proteins and try to correct that problem, try to promote the transition of that substrate into a native protein structure. So how does this work really? Well, the molecular chaperone, first, what it is overall going to do is take a folding intermediate, or hopefully maybe an aggregate, something like that, and it's going to try to lift those folding intermediates, those misfolded proteins, out of the local energy minima that I've talked about. So basically, imagine you have a protein that is misfolded, or it's a folding intermediate, and it's stuck in a local energy minima of that folding funnel that we talked about recently. So basically, your chaperone is going to step in and try to pull your folding intermediate out of that energy minima so that it can continue finding its way down the folding funnel towards the lowest energy native state. That is the goal. Now, how does this actually happen? Well, a family of proteins that are collectively referred to as heat shock proteins facilitate this process. So molecular chaperones, heat shock proteins, these labels are used interchangeably. Heat shock proteins are simply a class of proteins, a class of chaperones, in fact, that are overexpressed under conditions of thermal stress. So in cells, when exposed to chemical stress, thermal stress, what can happen is you start to stress out the proteins that are contained within that cell. And so under those conditions, cells overexpress these heat shock proteins in response to the stress, and they step in and try to ensure that proteins remain folded as long as possible. They're basically quality control enzymes. That's what they do. Now, the example that's being shown here, this is HSP70, heat shock protein 70. You'll see in a moment that the number here gives you a general sense for how big these enzymes are. So HSP70 is a very well characterized example of a molecular chaperone, a heat shock protein. So here we're going to use it as an example. So first, how does HSP70 and more broadly chaperones, how do they recognize their substrates? Well, the substrates that they're looking for are these folding intermediates, these aggregates, these misfolded proteins. The commonality amongst all of those cases is that you are going to have exposed nonpolar functional groups that would normally be solvent inaccessible in the folded native protein. So the exposure of that nonpolar surface is like a big red flag that says, hey, I'm misfolded, or I'm a folding intermediate. I'm not the native conformation. So in those cases, what's going to happen is your chaperone is going to recognize that nonpolar surface and bind to it. The nonpolar red flag, to use that analogy, is basically what the chaperone is looking for. It's going to bind to that nonpolar segment, and that is what is going to initiate the series of events that we're about to talk about. So HSP70, it exists in equilibrium between an open state and a closed state. So these are two distinctly different conformations. Unfolded or partly folded proteins bind first to the open ATP-bound form of HSP70. A second protein, HSP40, this is another heat shock protein, then interacts with the HSP70 ATP-bound, substrate-bound complex, and that's going to trigger ATP hydrolysis that produces the closed form of the complex. So here, ATP binds, protein that is misfolded, unfolded binds, HSP70 interacts, and that induces ATP hydrolysis such that you transition to the closed state over here. In the closed state, the domains here that are colored in orange and yellow, they come together sort of like two parts of a jaw. So what this does is it traps parts of the unfolded protein inside. You'll see here this unstructured region here, it binds, and now it's sort of bound within. It's on the interior of this complex. Dissociation of ADP and then recycling of the HSP70 requires interaction with another protein, a nucleotide exchange factor, for this to continue through multiple cycles. So for a population of polypeptide molecules that are unfolded, folding intermediates, basically the things that were recognized as substrates in the first place, only a fraction of these molecules are going to be, only a fraction of these molecules, when released after this open to closed transition occurs, are going to actually fully refold to the native state. In many cases, you're going to see that multiple cycles of this process are going to be required to fully remodel a folding intermediate into the native conformation. So here, for this first cycle, what might happen is, as this conformational change occurs, as you go from the open to closed state, you're going to basically induce a pulling force. So that conformational change is basically going to pull on the bound substrate, and hopefully that pulling force is going to cause some stress on the misfolded structure that is going to pull it out of that local energy minimum. But then what can happen is when a nucleotide exchange factor comes in to promote dissociation of the ADP, this substrate can dissociate, and it's, if not fully into the native state yet, it may rebind to another HSP70 and do the whole thing again. So my point in telling you this is I want you to understand that these proteins, these chaperones, are not 100% effective per ATP put in. Usually this requires multiple cycles, multiple ATPs to be hydrolyzed in order to fully remodel a folding intermediate or misfolded protein back into the native state. Molecular chaperones are involved in a wide array of different cellular activities. Here we're going to highlight just 4 representative examples, 4 families, in fact. And I want to, as I go through this, sort of highlight to you that this is just a subset of the different types of chaperones that are found in the cell. HSP proteins, heat shock proteins, they show up all over the place for different applications. So let's go ahead and just start. Let's talk about the HSP70 family. So first, let's talk about the name. What does it mean? You already saw HSP70 on the previous slide. You saw HSP40. What is this HSP and the number? Well, HSP just refers to heat shock protein. So this is heat shock protein 70. The 70 here tells us the molecular weight of the family as a whole in general. So most HSP70 family members are going to be 70 kilodaltons in size. The HSP40s are going to be 40 kilodaltons. Down here, HSP90s are 90 kilodaltons. There are also HSP100s. So the naming here tells you something about the size of the protein, the molecular weight of the protein. HSP70 proteins, as you just saw in the previous slide, function in concert with the HSP40 co-chaperone, and they effectively facilitate the folding of newly synthesized proteins and reverse denaturation and aggregation of proteins that were generated under stress conditions or just under the normal conditions that might promote that due to marginal protein stability. HSP70 proteins function in many different ways across the cell, which include, for example, unfolding of proteins in preparation for transport across membranes. And so they may induce unfolding so that proteins can be sort of pumped across membranes. So you'll see that the HSP70 family is critical to the stress response, but also in just normal compartmentalization of proteins as they localize to different parts of the cell. So trigger factors. So these are ribosome-associated chaperones found in prokaryotes that prevent aggregation of polypeptides as they emerge from the ribosome. So trigger factors and HSP70s are actually the first chaperones that a newly synthesized prokaryotic protein encounters. In E. coli, there is some redundancy here. You can actually delete the gene for either trigger factor or HSP70, but not both. They are functionally redundant. So if you delete HSP70, trigger factor can step in and keep things going. If you delete if you delete trigger factor but not HSP70, it goes the other way. You just can't get rid of both because they are functionally redundant. They are there to regulate quality control. They are also there to help proteins that are newly synthesized fold. That's what they do. Okay, so chaperone-ins. These are very, very large multi-subunit complexes that are found across kingdoms. They bind to improperly folded proteins and induce them to unfold inside of an internal cavity. Chaperone-ins structurally are very unique relative to the HSP70s and the trigger factors because they're basically a big giant barrel, and folding occurs inside of that barrel. You'll see in a moment what this looks like. We're going to focus on chaperone-ins in the coming slides. The last family here are HSP90 proteins. HSP90s are among the most abundant proteins in eukaryotes. They actually account for about 6% of cellular proteins under stress conditions. And they are also primarily involved in the late stages of folding for proteins involved in signaling. So they're really important for quality control, but they're also important for the later stages of protein folding. Most chaperones are going to require ATP. In the example where we talked about HSP70, you saw a very common example of how these things work. So the molecular chaperone is going to bind to an unfolded protein or a misfolded protein, protein folding intermediate, aggregate protein, whatever the actual substrate is, it's going to have a solvent-exposed hydrophobic surface. So the molecular chaperone is going to bind to that. And then, as you saw with HSP70, ATP hydrolysis is going to occur. That's going to generate a pulling force on that bound protein substrate, and ultimately, the goal here is to use that pulling force to induce denaturation of the misfolded protein or the protein folding intermediate and basically reset the whole cycle so that we can try to fold one more time. And so normally, this is what we're going to refer to as a bind and release process. So this is not going to, in most cases, occur successfully on the first go. You're going to bind, you're going to sort of tug on your protein substrate. That may disrupt structure, but it's probably not on the first pass going to fully unfold your protein. So the goal here would be to fully unfold your protein so that that unfolded protein can start to basically try to fold to the native state one more time. It gets another opportunity to fold. This is much more efficient than just outright degrading it and resynthesizing. So if you give that misfolded protein another opportunity to find the native state, you don't have to invest all of the ATP and all of the energetic resources into synthesizing it again. So this is just the more effective, efficient way to go. So usually remodeling is going to occur via a bind and release process that involves multiple cycles. Each cycle is going to involve one round or a round of ATP hydrolysis. You'll see that, like for HSP70, one ATP was hydrolyzed, but for chaperones, it may involve more ATPs that are hydrolyzed per round. But either way, ATP hydrolysis is coupled to the unfolding, or rather the remodeling process. And all of this, the role of ATP hydrolysis here is really to drive conformational changes in your chaperone that facilitate pulling on your bound substrate and hopefully then releasing of that substrate. We are now going to spend time talking about the best characterized example of a chaperonin. So remember, the chaperonin family, they differ from other molecular chaperones in the fact that they are large multi-subunit complexes. So here, we're going to focus on the GROEL-GROES system. Here you have a side view and a axial view looking basically down the axial channel of the GROEL-GROES complex. Now, okay, let's break this down. Each tier of the GROEL subunit, so here what I want to point out is that in green and red, these are GROEL rings. Each ring represents a 7-membered heptameric ring. So if you flip it on its side, there's a 7-fold symmetry axis. So you have 7 GROEL subunits packed in a ring, which pack against another separate 7-membered GROEL ring right here. So these two rings basically pack on top of each other to form what is effectively a 2-compartment system. On the top is a cap. So this is the GROES cap. It also has a 7-membered ring as well. So its function is to cap off this chamber. So what I want to sort of highlight here is that entry into, say down here, entry into this bottom chamber, you can't just diffuse in and then enter into the other chamber. So there's a barrier to allow diffusion between chambers. If you diffuse in here, you're going to go out the same entry channel that you came in. So you can't just diffuse in here and then move to the other chamber. It doesn't work like that. Okay, so the dimensions of this thing. It's nearly 200 angstroms long and about 140 angstroms across. And the entry channel, so you'll see the GROES cap here, leaves only about 10 angstroms of space. So it is a very good and effective cap to regulate entry into this channel, into this chamber, I should say. Remember, the substrate here that we're talking about, the thing that is going to be diffusing into each of these chambers, I'll tell you how it works in a moment, but the thing that is going to be diffusing into these chambers is going to be a misfolded protein or a protein folding intermediate. Usually these are going to be bigger than 10 angstroms. So this 10 angstrom hole here, it's not big enough to allow those protein folding intermediates or misfolded proteins to diffuse in or out. So the GROES component, the GROES ring that is shown in orange here, it is a cap to regulate entry and exit from the chamber that it is capping. So to give you a little bit of language, the ring here, the GROEL ring here that is touching directly the GROES cap is referred to as the cis ring. The GROEL ring here that is not contacting the GROES component is known as the trans ring. Okay, so you have here effectively a 2-chamber system. So you have 2 chambers in which protein remodeling can occur. Now let's talk about how this goes. Here we have the mechanism that describes the function of the GROEL-GROES complex. This is basically the series of molecular events that are involved in each cycle of protein remodeling. Okay, so let's start from the beginning. So top left corner here, you have, remember, a 2-ringed GROEL complex. So 2 7-member rings packed against one another to create 2 distinct chambers. A protein folding intermediate, so this would be one of your substrates, is going to diffuse into that trans ring, the ring that is not in contact with GROES. It's going to diffuse on in here. Okay, so now before I go any further, I want to highlight sort of what is going to happen. So this substrate is going to diffuse in, and it's going to undergo capture. So what do I mean by that? What? So remember, your folding intermediate is going to have nonpolar functional groups that are solvent exposed. So it's going to diffuse in. And at this stage, the interior of this chamber is largely nonpolar. There's a lot of nonpolar surface. And so via the hydrophobic effect, an interaction occurs between the nonpolar parts of your substrate and the nonpolar surface in your trans GROEL ring. Okay, so before I go any further, I wanted to actually show you what this looks like. So I pulled up the structure for the GROES complex. Here you have the GROES cap up here and the cis GROEL ring here, and you'll see that nucleotide is actually bound. There are actually 7 nucleotides. In this case, this is ADP, but could just as easily be ATP. 7 nucleotides bound in that cis ring. And then on the bottom, you have the trans ring. And here, no nucleotide is bound. Okay, so what I really want to show you is this. I want to show you the electrostatics. So what I'm showing you is an electrostatic surface of the GROES complex, and I've stripped away 3 GROEL subunits on each tier so that you can actually see to the interior of the chamber. Okay, so blue is equal to positive charge, red is equal to negative charge here. So what I want to sort of point you to is this is the interior charge distribution of the cis ring. So I want you to take note. You see a lot of blue and a lot of red, right? So there's a lot of hydrophilic surface in here. Now, if you move down to the trans ring down here, so remember, let me just sort of superimpose this for you. You can see here we have the trans ring, here we have the cis ring, and what I want you to see is that for that trans ring, there's a lot of white down here. There's, of course, some red and some blue, some charge, but white here. This is hydrophobic nonpolar surface. So in that trans ring where GROES, the cap, is not interacting, remember we have the GROES cap on the other end, on for the trans GROEL ring where the cap is not interacting, there is a lot of nonpolar surface. There's a lot of hydrophobic surface that is solvent exposed. This is not accidental. This is to facilitate capture of your folding intermediate, where you also have nonpolar material, nonpolar functional groups that are solvent accessible. So this folding intermediate diffuses into that trans GROEL ring where you have that nonpolar hydrophobic surface and undergoes capture. It is basically bound via the hydrophobic interaction that occurs. Okay, so binding of this substrate is going to trigger dissociation of your GROES cap from the other end, which then allows for ATP binding at the protein substrate bound end. ATP binding triggers a conformational change, which then allows for GROES, the cap, to bind on that end as well. So now we have a capped off, substrate bound, ATP bound cis GROEL ring that is ready to go. We have a substrate that's bound to that nonpolar surface. We have a cap so that it can't get out, the substrate can't get out, and we have ATP. So now, ATP is going to hydrolyze in a concerted fashion such that all 7 of those ATPs are hydrolyzed to ADP. This leads to a very large-scale conformational change in that cis ring. Now, okay, this is really very important to take note of because this transition that drives this conformational change, it's going to lead you back to this. So here, remember, if I show you the original structure, I have actually ADP bound to my cis ring. I have GROES up here. So this is a pretty good representation of the state that we are currently at in that mechanism. If I remind you, the charge distribution is now very hydrophilic. So upon hydrolysis of ATP, there's a large-scale conformational change that causes the hydrophobic material to basically become buried away, and the chamber itself becomes much more hydrophilic in nature. This is also going to accompany a mechanical force that is going to be applied. So remember, in this state, my substrate is bound to that nonpolar surface, and upon hydrolysis, it remains bound for a time to that protein substrate, but as it sort of turns, as the conformational change occurs, it applies a tugging force to that bound protein substrate. And the idea is that by pulling on this misfolded substrate, you're applying stress that is hopefully going to denature it. So while in this hydrophilic chamber, the idea is that you are sequestering the unfolded protein away from the bulk cytoplasm, the crowded environment of the cytoplasm, and you're giving it a safe space to try to refold without interaction with everything that's going on in the cytoplasm. Okay, so now we go from here to there, and basically this stage represents where we started. So we just went through a round of ATP hydrolysis, we tugged, we pulled on this substrate, and now another folding intermediate can come and bind to the other end, the trans ring of the GRO-ES complex. Same deal, it undergoes substrate capture, ATP binds, which triggers a conformational change, which kicks off the GRO-ES cap that was on that other end, so that GRO-ES can bind to this end that I'm highlighting here. So this basically starts the whole process again, but at this point, this trans ring that had the original substrate, that substrate can dissociate and perhaps has refolded to the native state or to another protein intermediate state. Remember here, this is reminiscent of the bind and release model, where this is likely not complete protein remodeling, is likely not going to occur in a single round. So typically only 5% of substrate proteins fold to their native state in each reaction cycle. So usually this is going to involve multiple cycles. But at this point, the substrate that bound to the other end can undergo the same sort of remodeling. So ATP is going to be hydrolyzed. At this point, ATP hydrolysis to ADP can lead back to where we started. Okay, so all of this occurs through conformational changes that are driven by ATP hydrolysis. Substrate binds via hydrophobic effect. ATP is hydrolyzed, conformational change occurs that pulls on that substrate and hopefully allows for unfolding and then refolding, and then that refolded protein can dissociate, or maybe the folding intermediate can reassociate with another GRO-ES complex. So one thing to know here, each GRO-EL ring can be defined by a certain volume. If a protein is larger than that volume, it's not going to fit into the chamber. So this places an upper limit on the typical protein substrate that GRO-ES, the complex, can remodel. So what it has been defined as is typically GRO-ES is going to interact with proteins that are in the molecular weight range of 20 to 60 kilodaltons. Now, for those that are interested in playing around with the structure, let's say that you want to play around with this and you want to get a feel for what this looks like. You are welcome to go to the protein data bank. You have been introduced to this at this point. If you search for GRO-EL, you're going to get a ton of hits, and you'll see they are pretty similar. They're basically they've been characterized in different combinations of states. So if you look at the actual cycle here, there are some structures that look like this. There are some that look like this, for example. So people have tried to structurally characterize the different steps in that pathway. And so this is just one example. You can search by PDB accession code 1AON, and that'll give you the ADP-bound GROW-EL structure. If you are curious, you want to read more, here is the citation. You can actually go click on the PubMed or DOI link, and it'll take you to the paper. And I've been perusing this. It's pretty interesting. It's actually one of the original papers on the GROW-ES complex. But it gives you a sense for how, the dimensions of this protein, gives you also a feel for the motions that occur. So they actually quantified, upon hydrolysis of ATP to ADP, what does the conformational change actually look like? And there's about a 90-degree rotation accompanied with a 60-degree change in position for one of those subunits. So they quantified this pretty well. And if you're curious here, it's all there for you. So feel free to do further reading if you would like. Before we end, I like to give this example. I like to highlight how physical chemistry has been applied to understanding protein folding biology. So if you think about what is actually happening in the cell, an unfolded protein is likely not interacting with a single chaperone. It's probably actually bouncing between multiple chaperones. So here I'm going to give you an example of how this has been modeled. And again, for those that are interested, I've given you the citations down here. You can search for these papers. They've done a very good job at quantitatively modeling this. So what they have done is basically looked at the periplasmic space of bacteria. So here we have multiple individually characterized proteins that are known chaperones or proteases for that matter. So just to give you a sense of what you're looking at here, imagine you have an unfolded outer membrane protein. Remember, your proteins are going to be synthesized in the cytoplasm, and they undergo unfolding so that they can be basically pumped across the inner membrane. That's what you're looking at here. This is a translocase complex that basically translocates an unfolded protein across the inner membrane into the outer membrane space, where at first it is an unfolded outer membrane protein. It is ultimately going to be incorporated into the outer membrane, so that's where the outer membrane protein language comes from, but it's going to interact with an assortment of different chaperones before it makes it to its final destination right here. Okay, so you've got different options here. And one thing I'll note is that they're not all chaperones. This thing that you're looking at right here is actually the DEG-P protease. So there are proteases in the periplasmic space as well that would quite happily come along and eat this unfolded outer membrane protein before it makes it where it's going. The other examples, other structures that you're seeing here are all chaperones and are essentially very important to ensure that our unfolded outer membrane protein becomes folded so that it can ultimately find its way into the outer membrane. Now let's talk about what has been done here. I'm always trying to build up physical chemistry for you and I'm trying to show you how it fits into biochemistry, and this is a very, very nice example. Hopefully you'll agree with me. The first step here in modeling, realistically modeling the process of taking your unfolded outer membrane protein to its native state involves mapping out all potential paths. So to give you a feel for what you're looking at here, your unfolded protein can, say for example, recognize the BAM chaperone, and BAM, it's converted to the native state. Forgive me, that was a very corny joke that I had to make. Alternatively, the unfolded protein may be bound by the SIR-A chaperone, which is going to take it to the native state, or the FKPA2 chaperone, which will take the same path, or SKIP3. So all of these are chaperone paths that take it to the native state. You'll also notice that there is a DEG-P path. I just mentioned DEG-P as a protease, but it's weird because it also has a chaperone functionality. So DEG-P could also function as a chaperone, or it could take it this way and just eliminate it via proteolysis. So here, each of these proteins, these chaperones or DEG-P, they have individually been characterized. So, imagine we talk about studying proteins in vitro. You have taken each of these proteins in vitro and done kinetic characterization to place rate constants and affinities on them to allow for a description of each individual path. Now, once you have all of that information together, you can start modeling this as a network, which is what is shown to you here. So the unfolded protein has multiple options, and honestly, this is going to be more of a probabilistic choice. Which path do I go? Some of your protein may go this way, some may go this way, so there's going to be a distribution of paths here. Okay, so based on an extensive literature search, the authors of that study were able to identify 43 rate constants that describe as much as possible the different paths that I just showed you to sort of build that network. So what they did was they built, using 43 rate constants, 22 ordinary differential equations. I'm showing you just 10 of them here. You'll see, for example, the rates of change of the concentration of unfolded outer membrane protein as a function of changing time here. You'll see terms for as many of those chaperones as possible that were just shown on the previous slide, where each of them are characterized by an individual rate constant. So here, this first equation would just express the concentration of unfolded outer membrane protein as a function of time. So that's a very important one. But altogether, you had 22 different differential equations. So they were able to take these 22 ordinary differential equations and treat them as a set of coupled differential equations such that they could perform computational simulations. So this is really cool because what you could do is actually control the concentration of each of your chaperones. Every single protein that I showed you on that previous slide, you could modify or control the concentration of the enzyme, and that would allow for you to recapitulate, to basically build up genetic phenotypes that have been experimentally determined in E. coli. So we can use the available rate constants, do some math, and actually make predictions that are supported by biological experiments. Really, really cool. Here we have the results of all of that math. So first I'm going to focus your attention on the left panel here. This is basically a plot showing where your protein is as it goes from the unfolded outer membrane protein state to the folded state as a function of time. So what I want you to see is that at first, as this simulation starts, it bounces around between these three chaperones, FKPA, SKIP, and SIR-A. It bounces around a lot. But then after, say, 5 or so seconds, it finds its way to a stable interaction with SKIP, but then it dissociates and winds up ultimately transferring over to this FKPA. But throughout the rest of the simulation, it just sort of bounces around between these different chaperones. In fact, you'll see in a lot of cases it bounces back to the unfolded state. So remember here, this is basically a spectrum going from unfolded outer membrane protein to folded, and the intermediate states are basically your protein binding to these different chaperones. You don't see anybody quite make it up to the folded state, and not during this phase at least, but you'll see the unfolded protein continually repopulate after interacting with different chaperones. Now this is to some extent sort of a probabilistic process. So the unfolded protein binds to the chaperone, the chaperone doesn't quite get it back, to get it to the folded state, so it remains unfolded, but sort of bounces around for a while until it happens to hit a conformation that ends on this FKPA chaperone and leads to formation of the folded state. So here, this particular outer membrane protein can be said to have a periplasmic lifetime of 54 seconds. So it took 54 seconds for it to reach that folded state. Now this is really interesting because you can use these data to actually quantify the average interaction times occupancy that describe the interactions between that unfolded outer membrane protein and each of the chaperones. Now, this is all very useful because what we have the ability to do here is take, for example, Surrey. Let's, in our simulation, knock it out. Let's remove the contribution from Surrey. What you're going to see is that the distribution of, the way this bounces around is probably going to change a little bit. And you can actually use that behavior to calculate the fold change in unfolded outer membrane protein plus aggregate compared to wild type. So this would be the wild type simulation. And if you knock out one of these genes, SIRA, SKIP, or FKPA, what you can see then is sort of how does the concentration of unfolded outer membrane protein change compared to the wild type simulation. So when you do this, if you knock out SIRA, you'll see a very significant increase in the amount of unfolded protein that hangs around. If you say, for example, knock out SIRA and DEG-P, goes up even more. But if you knock out both SIRA and SKIP, you'll see that the fold change is not quite as significant. So this is incredibly interesting because what the authors of this paper, what they were able to do is basically take these simulations, model it in the way that I've described, and via these knockouts, these genetic knockouts, and when I say genetic, this is the simulation to hopefully try to capture genetic data. What they are able to actually do is recapitulate the observations reported experimentally from genetic knockout experiments. So it is possible from genetic experiments, knock out a gene, and then quantify, in this case, quantify how much protein, unfolded protein or aggregate do you have in the periplasmic space. And what they have managed to do here is actually pretty well describe computationally what is observed in actual biological experiments. So I'm trying to get you to see here that we can use the physical chemistry, the rate constants, the affinities, to build up using the math a very, very strong predictive program that would allow you, so for example, you could, instead of knocking out a gene, just change the concentration. Maybe you underexpress it or you overexpress it. By doing that, you should be able to make very strong phenotypic predictions. So think about the strength of this if you were to go into human systems where you start to talk about aggregate formation leading to human disease. Perhaps you could use an approach like this to model the actual events that are taking place that lead to different types of human disease. The diseases that you might be interested in are summarized here. There are at least 35 different human diseases that are associated with the extracellular deposition of what would normally be soluble proteins to form insoluble fibrous aggregates. So I told you these protein aggregates, they are typically toxic to cells. And what can happen is that they are produced and they wind up aggregating, they wind up forming these insoluble plaque materials that lead to disease. So common protein or common known protein misfolding diseases include Alzheimer's disease, which involves the aggregation of amyloid beta protein, ALS, Huntington's disease, Parkinson's disease, alpha-synuclein being the wrongdoer there. You've got different encephalopathies. So this would be, like mad cow disease as an example of that. So there are many different neurodegenerative diseases that are, or also, well, let's just say neurodegenerative diseases that are, in fact, related to protein misfolding. A protein misfolding disease that gets a lot of media attention is Alzheimer's disease. So here, what you have is the formation of ultimately a filament structure that is going to be the amyloid filament that is going to be toxic. But let's talk about where that comes from. So we're going to start with the native amyloid protein. And what can happen is this protein can become misfolded or partially unfolded. Now, if you look at the structure of the native structure, you'll see there are 3 alpha helical segments and then 2 beta strands that form a very small beta sheet. Very small beta sheet. So this native protein can come unfolded, can become misfolded, and expose those beta strands. It can also expose the alpha helical segments that were formerly occupied in the native structure. So at this stage, misfolded proteins can interact with other misfolded proteins via a self-association process. So you'll see here that we have beta strands from neighboring molecules interacting. We have helices, alpha helices from neighboring molecules that are ultimately going to sit next to one another such that they will interact as this structure grows. So as that structure grows, you're going to see these fibrils lead into the formation of filament structures. And so here, most of the interaction is between beta strands that drive this. So this is an ordered aggregate. But there are also going to be some alpha helical interactions too that can occur. This is going to lead to the formation of an insoluble amyloid plaque material that undergoes deposition in the brain. So here you should notice that you have brain tissue and it's not supposed to look like this. It looks kind of like Swiss cheese almost. There are some holes where there should not be holes. And this is obviously not good for human health. Hence the reason that we consider this as a protein misfolding disease. The ability to form an amyloid fibril is not specific to just the amyloid beta protein that we have been talking about. You can see many other proteins have the ability to form these amyloid fibril structures as an alternative to their normal folded conformation. When this happens, most of these proteins that do this, they have a concentration of aromatic amino acids in a core region of a beta sheet or an alpha helix that in the native conformation normally would not be so solvent accessible, but in this alternative conformation becomes solvent accessible. So for this reason, there is an age dependence in the context of human disease. If proteins can do this, it is more likely to undergo stabilization if mutations occur wherein certain amino acids in these core regions are substituted with aromatic residues. So the substitution in these regions with aromatic residues make it such that it is easier to form the filament or fibril structure. Therefore, disease symptoms do often show an age dependence due to these random mutations like I'm describing. So as an example here, you can see 2 different possibilities. So in the case of the typical amyloid fibril that is driven by beta strand interactions, you'll see there is significant stabilization due to pi-pi stacking that occurs between aromatic residues that are in that core beta strand structure. There's an aromatic residue on each beta strand, and this facilitates packing in the way that I've described that gives the overall fibril formation. In this case, this started from the amyloid beta peptide, and this sampled a different conformation that ultimately positioned these 2 aromatic residues to form this extended beta sheet fibril structure in the case of Alzheimer's. So to give you another example, I found an example. There are many amyloid fibril structures in the protein data bank, And this is one example. So here you have two peptides. There's an aromatic group, there's some nonpolar composition. And if you look at the overall structure, you'll see that we form this very stable fibril where the peptides are interacting with neighboring peptides. And so this is an example of one of these fibril structures that forms for a protein that is not the amyloid beta peptide. This sort of thing can occur in other proteins as well. Now we are at the end of chapter 4. So in this chapter, we talked about everything from the peptide bond and the steric arrangements that the peptide bond can adopt. So peptide bond planarity, dihedral angles, Ramachandran plots. We talked about secondary structures that include the alpha helix, the beta sheet. We built secondary structure into tertiary structure and talked about quaternary structure, and then started talking about aspects of protein stability and protein folding. So the process of going from the newly synthesized protein to the native state, whether independently or by chaperone mediation. So we've talked about multiple examples. We've covered a lot of material in this chapter, but this marks the end of chapter 4. We will next move into ways in which the proteins interact with their substrates, their ligands. So that'll be the topic of chapter 5.